Hello everyone, welcome to ICGEB for the Genome Editing webinar. I'm joined by Serena Zakinia and Andres Muro, who are both group leaders here at ICGEB. And we're going to have 5 presentations on genome editing. The first 2 are really just general introductions. There won't be time for questions in between the presentations, so please just text us your questions and we'll try and answer them the best that we can at the end of the webinar. For those of you who don't know, ICGEB was founded under the auspices of the United Nations, but we subsequently became an independent international organization, and we're one of the few that actually do basic science research. So I think we should get started with the movies. The first 2, the first one is going to be by me, and the second one is going to be by Vittorio Venturi, who is a group leader here also that works on the interaction between plants and bacteria. Hello, welcome to the International Center for Genetic Engineering and Biotechnology here in Trieste, Italy. We are a scientific center of excellence, and we're here today for a webinar on genome editing. Genome editing is a new technology that allows us to do things very fast and very efficiently. Things that we used to be able to do before, now we can do much faster. And because we can do things faster and quicker, there's things that we could never imagine doing before that now are almost straightforward. So for example, if we wanted to correct a mutation in a mouse, that could take us a year to do before, and now we can do it in a few months or even less. And it's really been a revolution for basic science, and it will soon be a revolution in medical science, and especially how we treat people with inborn genetic disorders or other disorders that are caused by somatic mutations such as cancer. The easiest way for me to explain how revolutionary this is to basically compare it to editing a text document. So before, with the old genome engineering technology, it would be like having to edit a long Word document, and if you want to make a change, you have to read through the whole document and find every instance and then make that change. So for example, if I want to change this text into something else, I would have to read the document, find every example of that text, and then make the change. Oops. And hope I don't make a mistake like I just did there. And so that is very inefficient for me, because every time I want to make a change, I have to read the whole document, and it's very easy for me to make errors, especially errors in missing instances that my brain doesn't catch as I read the document. With the old genome engineering technology, we would have success in probably 1 in a million trials, which really limits the kind of things you can think about doing. With the new genome editing technology, we're probably having success in greater than 1 in 10 trials. Okay, so the way the new genome editing works is like using the word function find and replace. And this is what we want to replace. This is what we want to replace it with. And you're done. So it's really a massive change in how quickly we can do these kind of studies. You're going to hear 4 talks on how genome editing is being used here at ICGEB for basic science research.
Hello everybody, my name is Vittorio Venturi. I'm a group leader at ICGEB, at the International Center for Genetic Engineering and Biotechnology, and I study bacteria. So I'm a bacteriologist. And my work specifically, I'm very interested in how bacteria regulate gene expression, and I pay particular attention to how bacteria signal each other via cell-to-cell signaling, how they produce small signals to recognize each other and behave as a community. And I study these mechanisms in plant-associated bacteria, as I'm interested to see how plant-associated bacteria form communities. And also in parallel to that, in recent years, I've also developed an interest to develop plant probiotics using bacteria, because bacteria are part of the plant microbiome and they're very important for the health of the plant. So we want to devise ways to use bacteria, wild-type bacteria, to produce microbial inoculants, so farmers can grow crops in a more sustainable way. Thereby, using these microbial inoculants, we can help the plant acquire nutrients and fight pests. So to basically make agriculture more sustainable by reducing the input of fertilizers and pesticides. Now, coming to the topic of the webinar, I'm very happy to talk about bacteria because studying bacteria has been so important in molecular biology, in genetic engineering. If we think about the first revolution of recombinant DNA technology, it was made possible by the discovery of restriction enzymes in bacteria. So enzymes, these are proteins which walk along the DNA, and when they find a specific sequence, lots of different enzymes have been discovered. When they find specific sequences, there can be 4 base pairs, 6 base pairs, or even more, the enzyme will cut the DNA. And this basically led the way to molecular cloning and genetic engineering. Apart from that, also modifying enzymes have been discovered, things like DNA ligase, polynucleotide kinase, polymerases. These have all been discovered and then used for genetic engineering thanks to research in bacteria. And if we think also the polymerase chain reaction, another revolution that took place in the mid-80s, studying bacteria has been fundamental to develop this technology. So coming to the CRISPR-Cas technology, again, this is a beautiful example of basic research in bacteria through decades. So this is not like a eureka kind of research project that gave way. This has been the result of many years of research in bacteriology. Scientists have been interested for many years to study how do bacteria defend themselves from viral infections. Now, viruses that attack bacteria are called bacteriophages. And these are extremely common on Earth. In fact, they're among the most common things around. They're in the sea, they're in ponds, on Earth, they're everywhere. And what they do is they have some protein capsule inside, they have either DNA or RNA, they have a tail, they sit on top of a bacterial cell, and they inject their DNA and basically hijack the bacterial cell, use it as a factory to use all the components of the bacterial cell as a factory to make more bacteriophages. The end result is that the bacterial cell will lyse and will liberate hundreds of different virus particles. So now it's only common sense, and it's only bacteria are very smart organisms. They have evolved ways to defend themselves from these virus infections. And this is what scientists have been studying for many years. How do bacteria defend themselves from virus infections? And what they've discovered is that as you see in this diagram here, the virus injects its DNA, and through evolution, bacteria have been able to acquire very small pieces of the genome of these viruses and incorporate it in their chromosome. And you can see here these small little bits of colors which are interspaced by repeats. These are small pieces of virus DNA of different viruses. And this is like their vaccination program that they have. So they have a small piece of DNA that resembles exactly lots of different viruses. So they're vaccinated from when they're attacked. And what happens is that to the CRISP, this is the CRISP locus of all these repeats, which then in between the repeats are all these pieces of virus DNA. They are transcribed into RNA, and these RNAs are not translated. And then through different mechanisms, because several mechanisms have been discovered, these RNAs are then processed, okay? And they're processed, and then what happens is that these processed RNAs, which contain a piece of the repeat and a piece of the RNA, which the sequence is exactly the same to some viruses, they then interact with a protein called Cas9, and this protein RNA interaction makes a complex, and these guys are around the cell all the time, and this is their vaccination program. As soon as, if they are infected by a virus, and the virus is actually being, the bacteria is vaccinated towards that particular virus, the RNA in that protein Cas9 will recognize that piece, that virus, and then will recognize and hybridize, and the result is that Cas9 will cut it and degrade that DNA. So there you go. The virus can no longer infect the bacterial cell, and the bacterial cell will survive. Okay, so this research has been beautiful. It's been beautiful research, and I have to say, this research started back in 1987 when the first reports of these regions in the chromosomes of bacteria were discovered, and then through the years, the scientists have then discovered that these pieces of DNA are homologous to viruses. They then discovered the associated Cas locus. And especially recently, in between 2005 and 2011, two scientists, one from the US in Berkeley and one from Sweden in Umeå, they really made very fast, they've provided very fast results to show this mechanism. So, as you can see here, this history and concept of this CRISPR-Cas system, as you can immediately see, as soon as we discover something through basic research, then you immediately think of applications. Here we have, compared to the restriction enzyme that I talked about earlier, that were discovered in the late 70s, here we have a system where we have an RNA and protein together, which can take let's say the nuclease, the protein that cuts the DNA, to a specific region in the chromosome. And by changing that particular sequence that is recognized by the chromosome, here we have a very specific endonuclease which can do a double-strand cut at the region that we decide to. And what has been fascinating is that this very simple system, and rather cheap system as well, can then be used even for eukaryotic systems. So at the moment, as you will hear also in this webinar, the system can be used in genetic engineering or genome editing, as it is called, so making cuts in the genomes of different eukaryotic model systems. And we can cut a specific place, and then once it's cut, you create a mutation, or by repair, or even then you can then use biomolekular combination. You can introduce pieces of DNA that you want. So really beautiful genome editing, and that, it is another revolution. And obviously there's a lot of questions. The system has to be made more perfect, as we don't want it to make any mistakes. So there's going to be a lot of research and a lot of applications in the future. So I just want to finish by saying, how can we use this in bacteria? We in bacteria, we're very lucky. We have established methods of genome editing for many years. We in bacteria, we can do a lot of things already. We can mutate genes, we can put DNA in the chromosomes, or can replace DNA. So it's not going to be a big revolution for our genetic research, or also molecular biology of bacteria. I do think, though, that will be used in clever ways to do things that maybe it's difficult to do with established methods, like removing big pieces of chromosomal DNA, or maybe doing multiple mutation in chromosomes. So I'm sure it will be used in a very clever way to do exciting things, but it will not be a big revolution, because we bacteriologists are very spoiled. We can do a lot of genome editing already. Maybe also it can be used in some bacterial species or strains in which established methods are difficult to use. So I think the big revolution will be in eukaryotic research and in applications in various fields, in plant research and also medical research related to eukaryotic models, and also humans, of course. Okay, thank you very much for listening.
Thanks a lot for that introduction, Vic. Now we're going to go into a talk that's much more about applying genome editing, and it's going to be from Tanushree Kaul, who is a group leader at ICGEB New Delhi, and she works mostly on crop improvements. And so now we're going to have a taste of genome editing for agriculturally important crops.
Crops provide food, feed, fuel, and other consumable resources for human life, thereby contributing to the progress of human race and society. The world population is predicted to hit 9.6 billion by 2050, and globally the demand for crops will increase by 100 to 110% in the next 30 years. Thus, to feed and nourish the burgeoning population in the face of climate change, decreased arable land, and water shortages, there is a pressing need for innovation in crop breeding technology to increase agricultural productivity and accelerate sustainable agricultural development. Nature has been altering, editing, or rewriting genomes for thousands of years, with natural selection enabling organisms, both animals and plants, with genomic variants to survive. Moreover, humans have been employing artificial selection to domesticate crops for more than 10,000 years. The quantum of crops that we presently consume have been domesticated from their wild progenitors within the last 12,000 years. For instance, modern corn was produced by this process from its wild ancestor Teosinte. Crossbreeding, mutation breeding, and transgenic breeding are currently the main methodologies for crop improvement in modern agriculture, also referred to as first generation, second generation, and third generation technologies. Amongst these techniques, crossbreeding and mutation breeding are time and labor intensive and have led to genetic erosion or reduction in genetic diversity. Currently, fourth generation breeding technology is emerging as precision design agriculture via the integration of deep exploration on crop functional genomics, popular precision editing technologies, and big data mining that are synergized into breeding strategies. Amongst all the existent programmable site-specific nucleases, CRISPR-Cas system has explicitly emerged as prominently efficacious tool that enables precise and targeted genome modifications or alterations in diverse organisms, including plants. The mechanism of altering genomes is an offset of the innate ability of Cas proteins, especially Cas9 and CPF1, to induce double-strand breaks in DNA, in consonance with the short guide RNA molecules. The CRISPR-Cas tool has proven to be dramatically less cumbersome than meganucleases, ZFNs, and talons. Relevant plant genome editing tools, especially candidates from type 2 and type 5, have emerged as crucial edifices in the history of plant science and breeding. CRISPR-Cas9 or type 2 based system and Cas12a CPF1 are the pioneering reagents employed in editing of plant genomes because of their simple and efficacious nature of engineering and adaptability for multiplexing. One of the crucial issues to be considered while planning and conducting crop genome editing in plants includes the selection of target genes. Some traits, for instance grain amylose content in rice and oil quality in soybean, can be improved by targeting a single gene. On the other hand, crucial agronomic traits, for instance yield and biotic or abiotic stress tolerance, qualify the quantitative category that are regulated by QTLs, many of which have been identified, mapped, and eventually cloned, thereby offering an attractive resource of potential targets for genome editing in plants. The first approach is based on employing genome editing tools for gain or loss of function mutations for genes that are negatively influencing a targeted trait. This can be achieved via gene disruption, firstly by frameshift mutations that generate loss of function alleles that are indels, secondly amino acid substitutions that can generate partial loss of function or gain of function alleles, for instance via base editors, and thirdly, screening key functional sites in a protein via high-throughput mutant libraries. Genome editing can also be achieved through tools to regulate gene transcription via methods like generating random deletions in promoter regions, which can create allelic series with varying gene expressions. Secondly, targeted disruption or creation of transcription factor binding sites may cause predictable variations in gene expression. Further, non-homologous end joining or homology donor repair mediated fragment insertions in promoter regions also influences expressions. Moreover, changing of DNA methylation levels in promoter region via employing epigenome editing tools that may activate or repress gene transcription are also in the process. Narrow genetic diversity of cultivated crops is a prime cause for yield plateaus observed in breeding programs. According to a recent report, 120 essential rice genes regulating quality and yield traits revealed 28 genes having a single haplotype in a 3000 rice genome panel. Genome editing tools can reconstruct this genetic diversity in beneficial genes or alleles. Genes and QTLs identified as negative regulators of beneficial traits are the simplest targets for genome editing for loss of functional alleles in multiple crops, owing to their conserved nature. However, as umpteen QTLs are a subset of multiple developmental processes, so their modification may render negative effects. Selecting an appropriate genome editing approach is another factor that is important to accomplish the desired outcomes, as numerous genome editing tools are available to incorporate diverse modifications in crop genomes. Amongst the tools and strategies to modulate gene expression at the post-transcriptional or translational levels, mutating the initiation codon of the upstream open reading frames, for instance in lettuce and Arabidopsis, resulted in enhanced translation of downstream primary ORFs that led to increased ascorbate levels. Fine-tuning of the CRISPR-Cas system to alleviate off-target activity of Cas proteins can be achieved via optimization of guide RNA designing, engineering of Cas9 and CPF1 variants that recognize non-canonical PAM sites in the genome and simultaneously evade anti-CRISPRs, efficient DNA-free delivery of Cas9 proteins and RNP complexes through bialistic approach, enhancing HDR pathway frequency via inhibition of the NHEJ pathway, employing inducible systems to narrow down the availability window, using CRISPR genome-wide screens, and chimeric fusions of catalytic domains of deactivated Cas9 and FOC1 proteins, etc. Further, improving existing delivery methods or workflows and creating novel platforms for delivery methods and delivery targets. Strategic utilization of this ingenious CRISPR-Cas toolkit by researchers all over the world has effectuated targeted improvement of umpteen crop traits, quintessential for sustainable development. This technology has boosted the immune system of cacao plant to fight the virus ravaging West Africa, to produce fungus-resistant bananas and grapes, to generate more grains of corn and rice under drought, decaffeinate coffee and reduce the gluten content in wheat, anti-browning mushrooms and flavored tomatoes. Plant Biotechnology Division at ICGEB has successfully developed CRISPR-edited maize, rice and pigeon pea lines that are tolerant to glyphosate, a non-selective herbicide, and various crop lines with enhanced yields. Besides the development of glyphosate-resistant corn, rice and pigeon pea, with enhanced aromatic amino acid profiles, the nutritional improvement of crops team at ICGEB has also successfully generated CRISPR-Cas9 edited rice plants with enhanced iron and zinc content in their white rice grains. The Biofuel Division at ICGEB has engineered a synthetic butanol pathway in the genome of E. coli using CRISPR-Cas technology, and many more CRISPR-edited products are in the offing. By employing the multiplexed CRISPR-Cas9 genome editing approach, the Nutritional Improvement of Crops group at ICGEB is currently in the process of generating cereals with low phytic acid. Further, tomatoes with high lycopene content, a potent antioxidant and scavenger of free radicals that reduces the risk to cancer, as well as tomatoes with reduced allergenicity via reduction in the profilin, a small actin-binding protein that contributes to the allergenic potency of many fruits and vegetables, are forthcoming. India is the third largest exporter of groundwater in the form of rice and wheat. However, 21 Indian cities are predicted to run out of groundwater by 2020, affecting 100 million people. Human race is riveted on evolving and transmutating beyond fossil fuels as a strategy to adapt to man-made global climatic changes. Yet another frequently unnoticed climate change malefactor is the animal agriculture, which is the second largest contributor to man-made greenhouse gas emissions after fossil fuels, and a prime cause for deforestation, water and air pollutions, and loss of biodiversity. CRISPR-Cas technology has emerged as a redeemer with the potential to transform human lives via transforming crop genomes that can adapt to the inevitable global climatic changes in order to feed and nourish us. We at ICGEB are facilitating the redesigning of crop genomes via this CRISPR-Cas technology for precision crop improvement in order to rewrite the destiny of the existing and future generations of this planet. Recently, to ensure smart minimization of carbon and water footprints, Silicon Valley startup Impossible Foods, spearheaded by a biochemist from Stanford University, has employed the genetically modified yeast to produce a soybean protein heme in their burgers that mimics the taste of beef to offer an environmentally optimal, nutritionally safe beef replacement plant-based diet. On the contrary, the European Court of Justice issued a very different decision, which would subject the CRISPR-edited crops to the same strict regulatory regimen that the continent uses for GMOs. The big question is now, where are we? As sensible citizens of this planet, we need to decide our stance wisely for the peaceful coexistence of our children on this planet in the near future.
Thank you very much to Nirshu for that very provocative and also beautiful video. Our next presentation is going to be by one of the panelists, Andres Muro. He's a group leader here, and his research pertains to the relationship between gene function and disease and developing gene-based therapies for certain disorders.
Hello, my name is Andres Muro. I am the group leader of the Mouse Molecular Genetics group at the ICGEB. Our group focuses in the development of gene therapies, including the use of genome editing platforms. Next, I will present here a few examples to show the genome editing applications. The discovery of site-specific engineered endonucleases allowed the relatively easy introduction of desired modifications in the genome, triggering a revolution in both basic and biomedical sciences. This approach is being applied to a number of different fields, including the generation of animal models, development of innovative therapies, biotechnology, and agriculture. The first example regards the generation of animal models. The use of engineered endonucleases such as the CRISPR-Cas9 platform results in an increase in efficiency with a major reduction in time compared to the classical gene targeting method. In fact, to generate specific allele variants, fertilized eggs are microinjected with Cas9 selected single guide RNAs and sequence-specific oligonucleotides. Eggs are transferred into pseudopregnant females and mutant pups are born with a high frequency of mutated alleles. The whole procedure is very, very fast. Instead, the original gene targeting method developed by Nobel laureate Mario Capecchi, Martin Evans, and Oliver Smith was based on the spontaneous recombination of a targeting vector in embryonic stem cells, an event that occurs at very low frequency. Then recombinant cells are injected into wild-type blastocysts and embryos are transferred into pseudopregnant females. Some of the generated chimeras may transmit the target mutation to their progeny. This process requires a minimum of 1 year for a well-organized and efficient facility. Next, I will illustrate a few examples of their potential use in human medicine. Since not all allelic variants are deleterious and some provide protection to certain diseases, engineered endonucleases can also be used to alter metabolic pathways, obtaining a therapeutic benefit. For example, inactivation of the CCR5 gene, which is the HIV cellular receptor, prevents infection and development of the disease. In another example, the inactivation of the PCSK9 gene results in a decrease of plasmatic LDL and a reduction in the risk of developing atherosclerosis. Cas9 was also used to correct point mutations present in disease-causing genes, such as the one present in the mouse model of ornithine transcarbamylase deficiency, the most frequent genetic defect of the urea cycle. Other in vivo approaches are being tested in clinical trials, such as those proposed by Sangamo, using thin finger nucleases for Hunter syndrome and hemophilia B patients. In our laboratory, we have implemented genome editing approaches for the Crigler-Najjar syndrome, a rare genetic disease resulting in severe, potentially lethal hyperbilirubinemia. Spontaneous insertion of the therapeutic transgene downstream of the albumin coding sequence occurs in a low percentage of recombinant hepatocytes. However, this results in the rescue of the lethal phenotype and some of the disease features. Next, to increase recombination rate, we used CRISPR-Cas9 platform, fully rescued mutant mice, with bilirubin levels similar to those of wild type, with up to 20% of recombinant hepatocytes, elevated level of the therapeutic enzyme, and long-term efficacy. The efficacy of the use of CRISPR-Cas9 to correct disease has been demonstrated in a number of preclinical animal models. However, this approach still requires other developments, improving safety and immunological issues before they can safely reach the clinic. One main concern is the off-target activity of the nucleases, which consist in nuclease activity in genome sites that contain a sequence similar to that of the intended target site, as illustrated for the EMX1 gene. Potential mutations of key genes regulating cellular proliferation could result in tumorigenesis. Different strategies are being tested to reduce their off-target activity, such as dimeric CRISPR nucleases, optimization of single-guide RNAs, nucleases from other species, high-fidelity Cas9 variants, self-limiting Cas9 nucleases, and nuclease administration of lipid nanoparticles. So far, these strategies have been tested in laboratory conditions, and the conclusive evidence for their efficacy requires further experimentation. Another main concern regards pre-existing adaptive immunity against Cas9 proteins in the general population, as it may hinder the safe and efficacious use of Cas9 or cause significant toxicity. An important limitation of the genome editing approach by homology-directed repair regards the difficulty to modify non-dividing cells, such as neurons, hepatocytes, and adult cardiomyocytes, since the homology-directed repair machinery is mainly active during the G2 and S phases of the cell cycle. The molecular medicine laboratory at the ICGEB is addressing this problem by performing high-throughput screening of microRNAs that increase HDR in primary culture of cardiomyocytes. In another area, the CRISPR-Cas9 platform can be also used to control mosquito population transmitting serious disease, such as the human malaria vector Anopheles gambiae. The use of CRISPR-Cas9 gene-drive construct rapidly spreads the mutation in caged mosquitoes, while progressively reducing egg production to the point of total population collapse. Obviously, several concerns need to be answered before releasing mosquitoes into the wild. Gene drives have the potential to alter entire population and entire ecosystems. They could also, in theory, negatively affect human health by causing the malaria parasite to evolve to be more virulent or to be carried by another host. Researchers are developing gene drives with built-in controls, external overrides, or both, that should be unable to spread beyond a target population of mosquitoes or flies. The genome editing field has made enormous progresses in the last years, and more progresses are expected in the next future. Thanks all for your attention.
Okay, thank you very much, Andres. It's time for our last talk by another panelist, Serena. She's also a group leader here in Trieste. She works on the cellular and molecular mechanisms of cardiovascular development and disease. And she's going to talk about genome editing approaches in humans.
As we have heard, genome editing is a way of changing portions of a genome. We, as scientists, have been able to alter the DNA for more than 50 years, but now, more recently, this new technology allows to add, remove, or changing genes in a cheaper, faster, and more precise manner. A first distinction has to be made as to whether gene editing is applied to somatic or germ cells, as in the first case, the genetic intervention will only affect a few cells of the treated individual, whereas in the second case, the modification will affect all future generations. Researchers worldwide and at ICGEB are working to develop innovative therapies that use gene editing to treat a variety of conditions. This is not very different to what gene therapies have been doing over the last decade, with some major successes. In this context, gene editing wishes to change the DNA in cells of a sick individual to treat his or her disease. These changes are permanent, but since they occur in somatic cells, they only affect the person treated. One way this is already been doing in humans is by ex vivo editing of T cells to increase their ability to fight against advanced forms of cancer, including cancer of the esophagus, lung cancer, multiple myeloma, melanoma, and soft tissue tumors. These trials are currently running in both China and United States and wish to modify patients' immune cells by delivering a receptor particularly active in fighting against cancer cells, and at the same time using gene editing to eliminate a molecule named PD-1 that serves as a brake to the immune system and is also targeted by the new successful approaches of cancer immunotherapy. Despite some controversies on the appropriateness of the efficacy and safety of the procedure, similar approaches have been undertaken to treat some forms of anemia, in which CRISPR is used to increase the synthesis of fetal hemoglobin, as well as for the treatment of HIV, in which gene editing is employed to eliminate a receptor used by the virus to infect its target cells. In all these cases, some cells are harvested from the patient, gene-edited ex vivo, and then reinfused to the same patient. Other approaches aim at directly transfer the gene-edited tools in vivo to insert a specific DNA sequence in a precise genomic location. Sangamo Therapeutics is forging the way toward the use of gene editing for the therapy of hemophilia B, an inherited disease caused by the deficiency of a clotting factor, and for the treatment of two forms of mucopolysaccharidosis, in which the gene defect results in the toxic accumulation of complex sugars. In both cases, zinc finger nucleases are used to break the DNA in the albumin gene and promote the permanent integration of a wild-type copy of the mutated gene in liver cells. The first in-body CRISPR medicine testing has received its green light by the FDA in December 2018 under the name of EDIT-101. Editas Medicine and its partner Allergan are about to start a phase 1-2 trial in patients with a form of inherited blindness. This approach is based on the use of a viral vector based on the genetic engineering of the adeno-associated virus, or AAV, for the delivery of Cas9, as well as of 2 guide RNA molecules able to bring the molecular scissor to the site of the genomic mutation in the CEP290 gene, which is the cause of the disease, specifically in photoreceptor cells. There is a worldwide consensus to continue and extend the use of the technology to treat the vast number of patients affected by genetic diseases and also by complex diseases with a proven genetic component. This would require that the gene editing tools function specifically on the disease gene to minimize unintended consequences, and that the gene editing tools are delivered specifically to the cells affected by the disease, avoiding the other cells. On this line, the NIH last year has launched the Somatic Cell Genome Editing Program with the aim of expanding the number of gene editing tools available to researchers, developing delivery systems that can target the cells of specific organs and tissues in the human body efficiently, designing new assays for testing safety and efficacy of editing and delivery tools, and distributed the knowledge, methods, and tools that have been developed through this program to the scientific community. Over the last 4 years, some laboratories have started a more controversial use of gene editing in human embryos, eggs, and sperms. If an edited embryo is used to start a pregnancy, the changes would affect all the cells in the body of any resulting child and his offspring as well. A first paper appeared in Protein Cell in 2015, reporting the CRISPR-mediated editing of human zygotes in China. Following experiments in China, US, and UK confirmed that the technology can be successfully applied to human embryos to reveal the function of specific molecules during embryogenesis and also to correct pathogenic gene mutations. Until the end of 2018, when we were all shocked by the announcement of the birth of twin girls with edited genomes, again with the aim of eliminating the receptor used by the HIV virus to enter human cells. A lot of rumor is still going around this news, as definitive evidence that this has really happened is still lacking, and if true, the whole procedure has been conducted with no respect of standards. Finally, by definition, gene editing requires an in vitro fertilization procedure, which should not be allowed to people infected by HIV, as was claimed to be the case of the father of the edited twins. Dozens of countries prohibit any attempt to start a pregnancy with edited embryos, yet some scientists are encouraged to proceed. Gene editing poses a variety of societal questions which render it political and social justice issue, not just a scientific one. Yet, understanding the science behind genome editing is essential to formulate any ethical and social decisions. So far, most of the attempts to achieve an internal agreement split the discussion into 2 camps. Scientific experts explored technical issues, whereas scholars who study science and society address questions about the possible disruption to social norms. The 2 camps did not inform each other. To break out of this bifurcation between the science and the ethics, methods must be found to get people to engage with each other. I believe that a new type of conversation is needed to promote a wider, richer, and more complex knowledge of the technology and its associated risks and benefits within the society. CRISPR is more efficiently at precisely modifying the genome at a specific location than any other available tool, but there are obvious risks. CRISPR cuts the DNA once it recognizes a specific DNA sequence but a similar string of nucleotide might be present in other portions of the genome, and this could lead to off-target effects, where DNA disruptions may occur elsewhere in the genome. Yet this risk is difficult to quantify. We also know that genes in our genome interact with each other. Thus, would the editing of one gene indirectly affect the behavior of another gene? We can try to predict unintended consequences of genome editing, but there is still a substantial risk that some unforeseen events may occur. As with any medical intervention, we have to weigh up both benefits and risks. For instance, we all know that chemotherapy induces nasty side effects and kills also healthy cells, but we still administer it to cancer patients because we know that the benefits outweigh the negatives. A similar risk-benefit assessment will be needed for germline gene editing. In addition, this assessment also depends on the real need. Germline gene editing requires in vitro fertilization, and prenatal diagnosis already exists and is used to manage most of cases of potential transmission of genetic abnormalities.
Okay, thank you, Serena, and thank you to the other presenters. I don't need this. Now it's time for some questions. So if anybody out there has questions, just feel free to type them and we'll answer as many as we can. We already have collected a few questions and we're joined by Wendy Craig, who is the group leader of the biosafety group here at ICGEB. And as a matter of fact, she's here because the very first question is something that she can answer. So we have a question of what are the differences in the regulations on genome editing in different countries?
Well, I can only speak on behalf of the use of the technology in agriculture for now. So at the moment, most of the government regulation will be broken down based on whether they take a product-based approach or whether they take a process-based approach. So for example, if you're a country like the USA or Argentina in some cases, they will look at the, in this case, they will still call it a genetically modified organism, and they will look at it to see how different it is from the product that it's been developed to replace on the market. And they'll be looking at differences in phenotype and its composition, for example. And if there's no differences that would raise any concerns, then the regulation of it is relatively straightforward. However, the application of the genome editing technologies faces a much larger difficulty for those countries who take a more process-based approach, where because you are using biotechnology to develop the product, those resulting products will automatically fall under their national legislation. And because many of these genome editing techniques are still what are called emerging technologies, there's not a lot of products already available or being applied for authorization for release at the commercial level. So the most advanced countries are looking at those techniques that are very similar to traditional genome editing or genetic manipulation techniques like cisgenesis, where the EU, for example, will regulate those produced with this technique as if it were a GMO. Whereas other countries are looking at it and to see how different the application of the genome editing technique will be from traditional mutagenesis-derived plants that have been bred. So what I can say, there's no global consensus when it comes to the regulation of the products of these techniques when it's on a process base. But one of the key alerts or key controversies is going to be how will you be able to detect them and trace them through the marketplace if you're using a process-based approach, because In essence, they are technically no different from those that have been traditionally bred. So it's going to be very difficult, and it's yet to be resolved. So it's a wait and see stasis that we're in just now.
And perhaps in the other field, it is really important that since regulations are not in place, for instance, well, animal is more or less regulated by the possible applications to humans in a clinical context is still under discussion. So it's really relevant that people start meeting, and this is why as ICGEB we are organizing a workshop in the upcoming month to get scientists, regulators, governors possibly involved in the use and controlled use of genome editing from all over the world to meet and discuss, also to prevent major differences between countries and try to have a worldwide consensus. So in the meantime, I can read another question that is, what's the difference between traditional genetic engineering and gene editing? And I think Andreas can answer to this question.
Well, traditional genetic engineering involves normally the use of laboratory tools like bacteria, in that we modify sequences in the laboratory that then are used in cells, or they are using bacteria to produce a specific protein. Instead, the use of genome editing, we can directly modify the genome in the animal, in the organism, maintaining all the regulatory sequences and the regulation, the natural regulation of the gene, the expression of this protein or this system will be maintained. And this is a very important benefit for the study of the function, or for trying to let's say, in the case of the disease, just to try to cure a disease, maintaining the normal regulation. That this is very difficult using the traditional tools of genetic engineering.
And there is another question possibly for you, Andreas, which comes now. So how is it possible to be sure that the modification was only made in the specific section and was not made in other parts of the genome? Is there any long-term control that we can apply for that?
Actually, this question is very important, because this is one of the main concerns of the genome editing field, because once we introduce a modification, or we introduce a nuclease into an organism or into a cell, there are sequences that are similar to the target sequence that can be also modified. And in some cases, this can be deleterious, they can produce a tumor if some genes involved in the cell cycle are modified. And to improve this, we are developing very specific tools like high fidelity enzymes. We are also using modified modified Cas9, yeah, modified Cas9. We're also trying to deliver the nuclease in a very limited time window, in such a manner that the activity of the enzyme will be very short, reducing obviously the possibility of the enzyme to result in off-target. So this is an important question, and in the case of in vivo gene editing, it is very difficult so far just to be sure that everything will be in place, and this is the reason by which we really need to do a lot of experimentation to improve the technique and to reach a safety level that is enough to go in vivo in patients.
We have a few other questions. Some of them are related more towards the regulatory aspects, and some of them are more related towards the sort of the nuts and bolts about how it's done. So let's deal with some of the regulatory ones. And so we have two I'll just ask them and then we'll go around and answer them. And so the first one is it regulated that GMO organisms or GMO must be infertile? And then we have another one about regulation of the use of genome editing technology in humans, and is there any plan for regulation of this technology for the use in embryos? You want to answer the first?
Okay. So addressing the question with respect, should GMOs be infertile? Again, speaking from a plant perspective, this was quite controversial in the early days of GMO technology, and you may have heard of the Terminator gene, for example, where it was thought by the general population that this was a way for multinational corporations to control their properties and not allow farmers, especially in the developing world, to have free access to the germplasm and be able to develop it for themselves. So that type of infertility, built-in infertility, was very much seen as a negative aspect. So much of the development of GM crops, for example, have avoided this approach altogether. Now, however, there is gaining interest in this because of late, through the use of the application of the technology. For example, we heard about mosquitoes earlier, where some of the applications with the genome editing is to make the populations of mosquitoes sterile and thus suppress very quickly the numbers of mosquitoes that are around. So although it was controversial in the early days of GMOs that the products would be infertile, it's gaining in interest once again, depending on the application of the technology and how it will be used. So again, it's a case of watch and see for the time being.
Serena, I think, also has some insights into this question.
Yeah, so first question you asked was about the regulations on the use of gene editing in embryos. And this, of course, is not allowed by most legislations because we are not allowed to manipulate embryos in any sense. So this goes beyond genome editing. Even traditional gene therapy applications are restricted to somatic cells and not allowed in embryonic cells or germ cells so far. And this is, of course, reasonable, at least until we can control the technology. We can be sure that the rates of targets effect are very low and considerably low. And also, we should know what could be the consequences of the genetic modification. And this requires long-term studies on animals to be sure that changing an information has only the desired consequence and not major side effects. So for sure, there is a restrictive law, and it is good that it is in place. And I also see that there is another question about possible applications in the cardiovascular system. And there are a lot of studies in the case of Duchenne muscular dystrophy. Gene editing has reached almost the clinical phase. There are studies in cells from patients. And there is a group in the United States which is very close to the application in humans, where the deficit of dystrophin causes a severe form of cardiomyopathy. So this could be the first real therapy and use of genome editing for the heart. There are, of course, hundreds of other cardiomyopathies that are genetically caused. And so there is an opportunity for this. The vascular systems, the vessels, are much harder to treat because we don't have effective gene delivery systems to deliver the scissors and the guides to the cells of the vessel. So this is more difficult to think about now.
We have a number of questions related to like how you would go about doing genome editing in different kinds of organisms, or making sure that the genome editing is really efficient in different kinds of organisms. And maybe Andres can answer that one. So just in general, how would you go about
testing the efficacy
or developing in one that's efficient? I mean, how is it done now, for example?
Well, actually, now to modify other organisms, let's say or pigs or cows. Then normally this is done in the germline, so or even mice, as it was mentioned in the webinar. So normally what is done is introducing the nucleases, the scissors, plus the guides into the germline, plus a sequence that is a template to introduce the mutation, and then there is a recombination that occurs in the specific site in the genome, and then the animals that are born contain, a proportion of them will contain the desired mutation, and then this is the way to create a new population with the modification in the genome. And this is the real method that is normally used right now. And obviously, we should also control that there are not side effects or modification in other site of the genome. And to do that, one possibility is to do the sequencing of partial sequencing of the genome in the exons, and to try to verify whether there are mutations in these regions, and to do also transcription studies to check that there are no modifications in the normal metabolism of the organism.
Yeah, I just wanted to add that, you have to think about this as a really developing technology. It's not finished. It's not even what you would consider to be a mature technology. So there are improvements in designing of the guide RNA. There are improvements in delivering the guide RNA and the molecular scissors. There's improvements in the molecular scissors to limit the off-target effects. There's different molecular scissors if you want to modify multiple places at the same time, for example. I mean, so this is a field that is moving really, really quickly at the moment. And I would say that a lot of things now are being done as kind of trial and error to see which things actually work better than others. Okay. Is there we have another question, and this is probably the last one. And it is there anything known about the Chinese twins that were supposedly edited?
Yeah, eventually it came out that they were not edited at all, so they didn't have any genetic modification. There has been a little bit of rumor because at the same time, a paper came out in Nature Medicine saying that the same mutation that the Chinese doctor intended to create was shortening the lifespan in people. And so there was kind of concerns that the edited babies might have a shorter life. And this paper actually has been retracted today. There is the notification of the retraction coming out in Nature, so we can be sure that in the case they would have been edited, they probably would have a normal life. But still, any attempt has been so far not successful, and this again is an indication that we are not ready to go for the modification of the human germline, and we should wait until we have more efficacy and more safety on our side to proceed, if we will decide collectively that proceeding is good.
Okay, I think that's all the time we have right now. I would like to thank all the presenters and also all the participants who watched the webinar and asked questions. Okay, thank you very much.